Well, first, thank you, Jill, for the overly kind introduction. I appreciate that. And I'm just sort of the lead-off hitter here for an incredible group who's able to explain things. My task today is just to talk about what is cancer. And I think this begins with looking at what is defined as the hallmarks of cancer. And these were recently defined. One of them is self-sufficiency. They are, all right, I have too many screens. It's like these automatic driving cars. And if they work, they're great. But if they stop working, you start having extra crashes. And the cancer cells don't depend on other things the same way that normal cells do. And they resist the growth inhibitors. Our bodies are kept in check by control panels. And unfortunately, those don't always operate in cancer. Cancer cells resist death, much like Wonder Woman here in blocking the bullets. And they're immortal, like Yoda. They live forever. And people have kept these cancer cells alive for many years. One of the first ones was Gila, Henrietta. So that's an important characteristic. They induce blood vessels to grow in and support them. While they're doing that, they're evading the immune system, much the way that Harry Potter is putting on the cloak of invisibility. So the immune system doesn't recognize them. They have altered metabolism, like these two freaks. And they're genetically unstable so that they continue to evolve. So cancer, obviously, you're already seeing is complex, and then it adds the ability to move around the body and metastasize, and they promote kind of an inflammation. And I think one of the key points just to raise for all of you is that all cancers are defined as having the ability to invade, but not all invading tumors are able to metastasize. And this ability to metastasize is another characteristic of cancers. So how do cancer cells get these things? Well, if you look at all of those things, they're sort of lined up in the bottom. You can get from normal to cancer by a variety of different routes. You can tunnel under the wall, go over it, go through it, or around it. But the cancer cells change all of these things, and they do them in random orders. That's why each of the cancers that you all were describing has behaved just enough differently. But ultimately, what cancer is thinking about our normal bodies. You can think of it as a sink that's filled halfway, and you have the water going in and the drain going out, and those are in equal amounts. But what cancer cells do is they either turn up the spigot so there's more water going in, and the drain goes at the same rate, and what do you eventually get? An overflowing sink. Or you can clog the drain and have the same number of cells going in, and you can get the sink overflowing by either of those routes. So what can happen is the cells divide more or continue to divide when they should stop, or they don't die or stop growing the way you hope they do. And all of those changes are underlined by genetics or lifestyle, environment, or unfortunately, just bad luck. And the last one is something we can't control, but by looking at lifestyle and environment, maybe we can mitigate that a little bit. I put in this because I think it's really important to understand where we are today in understanding of cancer. And I like the history of what's been understood. 4,000 years ago, the Egyptians actually defined cancer, and they were treating it with chemotherapy and what essentially constitutes as immune therapy. Now, they didn't know that at the time, but they recognized that the cancer was a disease of tissues. And then with the development of a microscope, people began to look at the individual cells. And as we understood the cells a little bit more, new technologies allowed us to identify clinical markers, like I heard a couple of you talking about estrogen receptor or HER2 or other markers, and you're going to hear a lot more about those as the day goes on, so I'm not going to do it. And the last 10, 20 years, a number of other molecular genetic tests have evolved. And now we're beginning to define individual cells and the individual cells in our bodies by changes in cancer promoting genes, or which are also known as oncogenes, or lacking tumor suppressor genes. Those are like the brakes in the car. The oncogenes are pushing the accelerator. Tumor suppressor genes are pushing the brakes and stopping normal cells from growing. And those get turned off in cancer or lost or deleted. And then in the last half dozen years or so, another class of genes is evolving. And unfortunately, now when I show this to the medical students or graduate students, they ask what that third pedal is. Those of us who've driven a clutch car, a standard transmission, remember that we had to engage different gears. And these are what I call effectively modifying genes or susceptibility genes. Some people are more susceptible than others. Some races are more susceptible than others. And even with the same oncogenes and suppressor genes, you can have different speeds that you can go. And the cancer cells modify the behavior of those. So that's sort of what cancer is. I think the real question for this group is, why has it been so stinking hard to cure? And I think the bottom line is that cancer cells really don't do much that some normal cell doesn't do, but they do all of those bad things that I mentioned in combination. They have to have all of them to behave that way. And because of that, when we're targeting cells that are dividing, well, the normal cells that are dividing get affected and the side effects exist. And that's why we have what are called off-target effects or dose-limiting toxicities. And you're going to hear more about that from my clinical colleagues who will be able to talk about that. Another is what's known as tumor heterogeneity. Almost all cancers, well over 95%, start from one cell that's gone rogue. But by the time they're diagnosed, which is about roughly 1 billion cells, the composition of that mass, that tumor that exists, is tens of thousands of subpopulations, and they all behave differently. I don't mean to depress you by saying that, but that's why it's been hard. And because they're genetically unstable, you get rid of one population, the others mutate and cause problems. So it becomes an evolving target. And ultimately, most of the challenge with all of those things is when the cancer cells move and they move into different spots or metastasize. That's sort of what I like to study most. And that can occur by a bunch of different routes in the body via the lymphatics, across blood, through the bloodstream, or even across body cavities. And that is becoming increasingly important. I'm just going to skip that one. But I want to end with a little bit of positive, more than just a little positive. There has been progress, and you'll hear that from others. But back when I started graduate school, the prediction was any woman diagnosed with metastatic breast cancer had a 0% chance of living 5 years. That was what they were publishing for the American Cancer Society. Well, the 5-year survival rate overall now is closer to 45, 50% for metastatic advanced breast cancer. And that number continues to rise over years for all the cancers. So we have made progress, not as much as anybody would like, but made progress. So with that, a couple of take-home messages. because Sarah is not very nice in giving me 10 minutes to explain what it's taken me 40 years to understand. Cancer is a lot of different diseases. It's really complex. It's evolving. What we know today isn't enough, but we'll know more tomorrow because we got a whole team of people throughout the country and the world working on it. And you as advocates are helping shape the direction and the pace of research. I'm excited to be working with you as part of Pivot and look forward to continuing to do so. I did 18 seconds over, Sarah. I hope you'll forgive me.
Danny, you did great. Thank you so much. Danny's an expert in metastasis, like he had mentioned, and I can't believe you could distill all that in 10 minutes. So great job. Letty, do we have any questions in the chat before we?
No questions so far, but from me, thank you for the Harry Potter reference.
All right, well, we will jump right into actually to me. I get to talk next before I pass the baton. So I just wanted to provide a little context as far as how the research spectrum works. So we used to call this the march of science, that science would begin in the basic realm of preclinical and animal studies and then march along this continuum. Well, it turns out that cancer is complex, so sometimes things aren't quite so linear. But I do think it's helpful to understand that there's lots of different types of experiments and studies that are done. So the T0 of basic science and research, and then it matures into translation to humans, where we do really small studies, and then we translate that to patients in contexts where they're actually have cancer or taking treatments for cancer. And then it matures into translation into practice and studying clinical trials and clinical outcomes research. And then eventually we translate into the community at population level outcomes research. And what you'll hear today are from different program leaders that oversee aspects of different components of this continuum, and they will mention that. So that gives you kind of a task to do, like a treasure hunt, is to figure out which of these different buckets they actually are serving with their different researchers and their programs. And then I think the really key thing for each of you to understand is that your voice is critically important in each one of these, even the basic science research, and even more so sometimes the basic science research. We have some really cool partnerships already of some of our patient research advocates helping with things like muscle wasting and inflammation, but it's in Petri dishes and animal experiments. And it's been critically important for those basic scientists to hear the patient perspective. So I just wanted to provide a little context. So now that you understand cancer, that's how cancer research works in a nutshell. And I'd like to pass on to Dr. Kristi Neufeld. She is the Cancer Biology Program Leader. And Dr. Neufeld, are you ready to go?
Yep, sure am. Thank you, Bill.
Awesome.
So it's great to meet with you and talk today about the Cancer Biology Program. And Danny took this slide out of his slide set, so this maybe will make a little bit less sense, but I was borrowing one of his images from a picture from one of his slides that showed the progression or development from a normal cell, which is shown here on the left, is this nice little circle with a little darker circle in the middle, to sort of a less normal, more abnormal cell, and then the cell is kind of growing in an out of control manner. It starts to break off and move to new sites, and now maybe it's metastasized to the lungs, small metastases, and then larger metastases. And if you can click the next slide or the next, thank you. So cancer biologists are interested in this progression. How does a normal cell move to become a little bit abnormal and a lot abnormal, and finally ultimately become a cancer cell, and then a cancer cell that can metastasize and move to new locations? We're interested in each of these individual steps because each of these movements from one stage to the next involves different processes in the body. And so members of my program, research program, are really trying to figure out all of these question marks. What is the underlying biology that moves the normal cell to become a cancer cell ultimately. And the idea, the big picture is that we can, if we figure out all of those steps and what is involved in making normal cells become cancer cells, then we can use those to target therapies toward those specific events. Really big picture, again, looking at a normal cell and then a cancer cell, there would be differences maybe. And if we could figure out those differences, something that's only happening or mostly happening in the cancer cell, then we could target that therapeutically. We could also use that information to think about ways that we could prevent cancer, possibly. We could also use that as ways to diagnose cancer. And so really the science in the cancer biology program, as the title of the program, Cancer Biology, suggests, is studying the underlying biology of cancer. And this is basic science. So can you advance to the next slide, please, Sarah? So Jill showed this already, but on the research continuum, we're pretty much way over there on the left side of the basic science research. The way I describe basic research and how this fits into the whole picture of this pipeline of research that isn't really linear, I think of this as, so let's just imagine that you have been served this, one of your family members or friends has made this delicious meal for your birthday. And you eat this meal and you're so thankful, you're so grateful, and it's just delicious food. And you're saying, thank you for making this food. That's just awesome that you did that. And you thank the person that made you the food. But you don't usually think about all the steps that had to go into actually getting that food on your table. And so you can go back a little bit to the fact that there needed to probably be purchasing of the food items in the grocery store. Before that, there was probably some transportation and trucks that needed to bring the food to the grocery store. And before that, even at the very beginning, there were farmers and ranchers that grew that food, planted those plants, grew those animals that ended up being the food. And so I think of the cancer biology program members, the people in my research group, as being those farmers and ranchers. We're trying to provide that very first bit of information that is required, absolutely required to get to the end goal, which is that therapy at the end. But if you don't have the first information, if you don't have the basic components and you don't understand those basic components, you cannot get to the end. You can't get to a new therapeutic. You can't get to a new prevention strategy. And so the basic research is critical for that, because if we don't understand what's happening, we can't develop the therapies. But it's also a little bit behind the scenes. We don't get a lot of the sort of the PR and the flash that maybe the chemists or maybe the people that are doing the drug discovery or the later stages that we found a cure for this or we found a therapy for this, because it's really one of the very, very early steps for this. So the program is the cancer biology program. A little bit of the specifics about the program. We have 4 different program co-leaders, and part of that is because we represent 4 different campuses that are all part of the University of Kansas Cancer Center. And so I am located here in Lawrence, Kansas at KU, and Sufi Thomas is located at the Med Center, KU Med. We have Tomo Iwakuma, who's at Children's Mercy, which is a hospital and research center that, as the title implies, is predominantly focused on treating children's cancers. And then we have Lin-Heng Lee, who's at the Stowers Research Institute. And some of you might have heard of this institute. I think it's worth pointing out a little bit about its discovery. So Jim and Virginia Stowers, they both were cancer survivors, and they recognized quite a long time ago that this idea that really basic research was the driver of innovation. And if we don't have very, very basic research, then we can't have any sorts of drugs that are targeting cancers because we need that information. And so the Stowers Institute is really devoted to this concept of basic research. And again, the overall goal of the basic research is that we can take the things that we find in our labs and pass them off to probably the next group, or to the chemists and other scientists, researchers in the drug discovery and experimental therapeutics program, which is going to talk later on. And we pass off our information to them. they can identify drugs that might target the different mechanisms or the different components that we're finding. And then they might pass that information back to us again in the cancer biology group. And we might then study those compounds using cells in dishes grown in Petri dishes, or even in some animal models, and then pass that information back to the, back to the drug discovery and experimental therapeutics group. And then they further refine their compounds. And then eventually this hopefully will get moved all the way to clinical trials, that next stage. But there is a lot of back and forth between the different research groups. So where patient advocates, could you advance or click? Yeah. So where patients advocates can come in on this continuum, or in terms of helping us, is a lot of things. If you go back one slide, just one click. Yeah. So you can see that I added this arrow here from T2 all the way back to basic science research. So there is information coming back, hopefully, from the patients to our group. So if we can have the input of patient advocates, then that can help us in our basic research. Okay, now, sorry, for the next slide. So what specifically might you all be doing to help and to partner with us and collaborate? So our cancer biology program has about 100 members. So that's 100 different labs. Some of them have maybe just a few people in their labs. Some of these labs are quite large, but most of our labs will have a regular sort of meeting where we talk amongst the lab members about our science. And one of the really nice things that a patient advocate could do would be to attend our lab meetings or come to a lab meeting even and share your perspective with us. And learn about our research and really start to interact and talk about what it is that we're doing in the lab.
Would there be any desire to ask the question why, or is there a way to do that at this point to get some of the rationale on both ends of the spectrum, both those that were quite positive and those that were not as positive?
Is that a question for me, or should I just keep going? Sorry. Okay, I'll just keep going. So that a patient advocate could attend our lab meetings and therefore inform our research and learn about our research, and that would be great. One of the things that we need to do to keep our research going is we need to get funding for that research. And so we typically will need to write grants and submit proposals that said this is what we're going to do in the future. And we would like to get federal funding, ideally for that research. And patient advocates can really help write those grants and help inform what we're writing in the grants. And in fact, some of those grants now require what's called an abstract, which is just a summary of everything that's going to happen in that grant. But we need to write this in a way that anyone could understand this. So I could go off and tell my mother about this and she would understand that. And that's called a lay abstract. That's kind of point number 4. So patient advocates might be able to help us create those lay abstracts and bounce ideas back and forth about that. And then, as you can see on the slide, you all can read the slides, but there would be a lot of opportunities for collaborating in presentations at meetings and conferences, and really just overall consultation about our research and how it can impact you all or patients in general. With that, I think I will end and try to keep us on schedule. So,
okay.
Thank you.
Great. Thank you, Kristi Neufeld. I'm sorry, when I started the meeting, I didn't mention that we would pause for chat questions between each speaker and then at the end. And so I apologize, Broderick, I didn't mention that before. Regarding COVID testing and all of the rules with visiting, I know several labs are meeting by Zoom, and that might also be an option, as far as kind of navigating the pandemic. Letty, I just want to take a moment and ask, are there any questions in the chat before we go on to Dr. Dan Dixon?
No questions in the chat yet.
Okay, all right, Dr. Dan Dixon, are you ready to go ahead and?
Sure.
Okay.
How is everybody today? I know a few of you, and over the years, it is wonderful to see how much this program has really blown up and become really and I'll show you guys later, an integral really a central part of our research program and of the Cancer Center. So I really am happy to see where things are going with Pivot. So I'm Dan Dixon. I'm a professor here up at the Lawrence campus. I used to be at the Med Center, and I'm involved with the D3ET program, which is Drug Discovery, Delivery, and Experimental Therapeutics. Next slide. And so what our program is we have a number of members involved, and it's probably half to maybe 2/3 clinicians, and then the other program members are basic scientists, technology people involved with technology development. And you can see our leadership within the program really tries to cover those areas. So we have John Taylor, who is a surgical oncologist focusing in bladder cancer and other GI types of cancers. We have Priyanka Sharma, who is a medical oncologist. Her area is in experimental therapeutics, primarily in breast cancer. And then myself, and I'm a basic scientist. I work in the areas of high-risk cancer prevention and early detection and drug discovery. And so our combined leaderships really try to work together and represent the whole program. Also within our program is our associate director, Scott Weir. Many of you know Scott. He is very actively involved with the program and the number of different areas in the program that we reach out into. Okay, next slide. So you've already seen a version of this slide, and you'll probably see it all the way down to the end. This slide really sums up part of what we do within our program, within the D3ET program. And so if we look at this process of cancer drug development, this is something that, first of all, I can't point onto this, but you can see that this is not something that happens overnight. I think things, as it was alluded to earlier, things have become accelerated. The process of developing a drug, especially with the lessons we've learned through the COVID vaccine development, can be accelerated to benefit patients. But typically, things take a long time, and they happen in different stages. And you could probably divide this up into 3 different stages. And the stages that our program is primarily involved with are this first stage, drug discovery and preclinical testing. And then in the second stage, which is clinical trials. And that's where we look at how a certain drug would affect people in phase 1, phase 2, and phase 3 trials. Okay, and I'll give you some examples here of just what our program is doing in this process. And so first of all, one of the things that's central to our program is drug discovery. And so say we work with people in the cancer biology program that Dr. Neufeld highlighted earlier, and they found a certain protein is overexpressed in a tumor type that they study. And they may come to us and say, hey, we want to look for a drug that might be able to target this protein that's only in the tumor and maybe halt the tumor from growing, because they've done a lot of bench science to investigate that process. And so we utilize one of the things that's available to us in drug discovery screens. And maybe a number of compounds are identified, which with our experts in drug discovery and medicinal chemistry, those compounds get weeded down to maybe 5 compounds. And then those compounds move into working with the clinicians in our program into phase 1 and then phase 2 trials. And then ultimately, if things keep moving forward, our program is also involved with working with industry partners where things move into a much more expensive phase 3 type trial and then approval by the FDA. And so we have a lot of partnerships with industry and for-profit ventures. And so we really try to span this aspect of drug development. And so a little story, I remember when I was a postdoctoral fellow, I would go to these meetings, and some of them were very large meetings like the AACR meeting or a big gastroenterology meeting. I studied colon cancer. And I walk around on the vendor floor and I'd see these drug companies with these big charts of where they have drugs in a certain pipeline here. And I always be amazed at, wow, they're really moving things forward in this aspect. And maybe some of you have had a chance to go to meetings and seen those big charts, what these major drug companies are doing. Well, you know, they're not alone in this process, because if you go to the next slide, we are doing this also at KU. And this is just an example of some of the drug development that we have within the whole cancer center. And so we can focus on different target cancers, and some of them you might be familiar with: breast cancer, colon cancer, bladder cancer. These are the targets or the leads. So these are proteins or genes that are somehow altered in that cancer. And so they are the target that we try to identify drugs or small molecules that can somehow inhibit that gene or that protein. And that's a hit generation. And then we take that drug and we try to make it better. We make it last longer. We make it more effective. It gets to the tumor better. And then a lot of this part up here, these first 3 steps are really done at the bench and in more cell biology conditions. So then we have to take this stuff into an animal model and try to show that, okay, it actually works in animals that have tumors similar to the humans. And so this is the early steps, that first box that I demonstrated to you. And you can see we have a number of compounds or a number of studies that have reached this point that are now going to move on into people. And can you go to the next slide? And so that's where we have a number of different targets and a number of different compounds that have moved from the preclinical phase into the phase 1 and phase 2 steps. And you can see here, things are a little bit more better defined as to what is being targeted here, and then what the candidates are. And really, we're really happy with some of the stuff that's happening here that's moving forward into phase 2. And over here, you can see some of the bars here are light blue and some of them are dark blue. Well, the light blue are repurposed drugs. So these are drugs that may be used for asthma, may be used for antifungal agents. Well, some smart people, some smart basic scientists said, maybe this could work for a cancer. It's just never been looked at. And so we moved forward in that direction and repurposed an already known drug for cancer purposes. Other ones are drugs that have been invented by KU. And that's one of the really exciting things. So next slide. I'm going to go over here just a second because I wanted to demonstrate to you that really Pivot is in the center of our work. And our program here is involved with a lot of these different groups. And we are directed by the COE, which you're going to hear about in a little bit here, and then also your voices. So this is something that we're very passionate about, working with your group in the rapid reactors and some of the other areas that we can be educated from your perspective in how to best move that pipeline of drug development forward. Okay, thanks.
Dan, I really like this graphic and how you make pivot like the nucleus or the brain or the heart of the work. That's really cool. Thank you for sharing that. Hey, Letty, do we have questions in the chat?
No questions so far.
All right, well, we are so far on time. Super impressive, everybody. Dr. Tara Lynn is next, and she is the medical director for our clinical trials office. Tara, Dr. Lynn, are you ready?
I'm ready. Can you guys hear me?
Yep.
Great. Good. I just want to say a special thank you. This is always a wonderful group for us to work with. And this is my favorite topic of all time to talk about. So I will do my best to stick to the 10 minutes. Next slide, please. So when we think about clinical trials and where they fit into this whole idea of drug development and bringing new ideas with the goal of bringing new and better therapies to our patients, right? Our earlier speakers have kind of talked about where this part falls. And so I want to take some time to actually talk to you guys about what clinical research is and where it fits in. So these talks are very well designed. The way this agenda is scheduled to go from Kristi to Dan, and we have an idea in the lab and we've gotten some good preliminary data. And then where our team comes in is really when it's time to start to bring some of these new ideas, new strategies for treatment, and actually take them, we call it taking them from the bench. So taking them from the lab, taking them to the bedside so that we can actually start to make an impact for individuals. Next slide. And when you think about clinical research, so clinical research is when we're studying something new in people. So it's the difference between laboratory research where we're testing ideas, but on cells or on animals. And now when we're into clinical research, we're really making the intervention in people. And so when we think about having a comprehensive program, which is really what we strive to do, you think about all of the facets of cancer care. So that goes from cancer prevention, cancer screening in order to catch cancers earlier and hopefully be able to improve outcomes. I think what most of us think of when we think of clinical research is treatment, right? So testing brand new medicines, old medicines in new combinations or new dosing schedules. But then there's also a lot of clinical research in cancer care that's around the idea of supportive and palliative care. So whether this is interventions on supportive things like nausea medicines or quality of life surveys, and then even when we think about palliative care, symptom management, early intervention of the palliative care team versus later intervention of calling the palliative care team, those folks who are really focused on symptom management, sometimes focused on end of life issues. When we think about comprehensive clinical research, it's not just treatment and a new chemotherapy or a new surgical procedure. It really encompasses everything from preventing cancer and screening for cancer, but all the way through that treatment spectrum. Next slide. So why do we need clinical trials? And I think it's important to remember that the FDA approval for new agents and for either for brand new drugs or for older drugs for new diseases comes from the results that we learn from clinical trials. Next slide. So like we just talked about, right? So new screening interventions. Should we use a chest X-ray or a CT scan to look for lung cancer in patients who've had a history of cigarette smoking? When we look at cancer prevention, does this medicine or that medicine or no medicine work better to try to protect people with, say, a family history of a kind of cancer? Common ones that we think about would be chemotherapy, new surgical techniques. Should we take out this many lymph nodes or that many lymph nodes in someone with a cancer? When we talk about immunotherapy, this is this idea of new medications that really harness the immune system to help your body fight the cancer itself. All need to come to FDA approval through this clinical trials process. But then again, things like antibiotics, anti-nausea medicines, all of the interventions that we do, for the most part, are going to come through this clinical trial pathway. Next slide. So when we think about a clinical trial, it takes a lot of people, and there's a lot of effort that goes into building and designing a trial even before it gets to the time when we're presenting it to an individual. And so that includes the laboratory folks who helped develop the new idea, the statisticians who helped us design the trials to make sure that we're testing the right number of patients to be able to see an effect. And then when we talk about who is actually involved in some of the mechanics of working with individuals who are on trials, obviously the number one most important part of the whole process is the participant. So that's either someone with cancer in our discussions, right, or someone maybe with high risk of cancer that we're testing these new interventions. Many of our clinical trials come from a sponsor. So that can be a drug company or a device company if we're testing a new kind of implant for something. The federal government is actually the sponsor of many different kinds of research trials. And then we also have, like Dan was just showing, a nice portfolio of clinical trials ideas that have been developed here at KU. And so there are times when our own institution will serve as the actual trial sponsor, and that's kind of the administrator of the whole process. We have a lot of people who will interact with the study participant. There's a study coordinator and a research nurse who are actually there with the patient day to day while you're getting the study treatment or intervention. We have pharmacists within our department who are only focused on giving investigational medicines. So while they're, credentialed in all kinds of chemotherapy, it's this extra layer of investment and detail because we're giving, in some cases, brand new medicines to people and really learning about their side effects and how they interact with other medicines. And so the research pharmacist is really a very important part of our team. We also have, in many cases, a research lab. So for lots of cases when we're studying new treatments in people, we will need to do additional lab tests. So taking extra blood work and things to try to understand what are the drug levels in different people? How does this drug interact with a different drug that the person is on? And so having a dedicated research lab, within our site at KU really helps us to be able to do things. There's the physician who is involved with the clinical trial. For every trial that we have open at KU, we have one physician who takes the lead, but then certainly all the members of our team, for example, on the breast team, there'll be one breast oncologist who is the kind of in charge of the protocol at our site, but lots of different of the breast physicians would also participate. We also have a layer of monitoring and quality assurance to make sure that when the protocol says we need to do X, Y, and Z in that exact order, that all of us on the ground are doing X, Y, and Z in that order. Because the number one priority, our goal is absolutely to bring the best, most innovative, better treatments, but patient safety and our participant safety absolutely has to come first. And so that starts from the very idea when a clinical research protocol is, when the idea is very first starting, safety is built into it. The FDA is very involved in clinical trial development to make sure that all the safety pieces are in place. We have safety arms within our cancer center and within KU as the university who are also double and triple checking to make sure that patient safety really remains paramount in our minds. Next slide. I want to just give you a couple slides looking at kind of the scope of our program. So over the last 5 years, 2016 to 2020, we had over 13,000 patients involved in clinical research studies. So some of these were for new medicines, which is the one that you think of most often. Some of these are more observational, and some of these are also lab-based. Next slide. And when we're talking about this term that the National Cancer Institute gives us, this idea of interventional treatment trials, these are trials testing new medicines, new radiation approaches, new surgical approaches. And so from 2011 to 2015, we had just under 2,000 patients who came onto clinical research trials with us. And then over the most recent 5 years, from 2016 to 2020, we're over 3,500. So with the support of our community partners, the team at KU, everybody really working together, we've been able to make this tremendous impact on patients across our area. Next slide, please. I want to highlight for everybody our Clinical Trials Finder app. So this is available through the Apple Store or the Android store. And what it does is it walks you through several steps so that if you or a family member or someone needs to figure out what kinds of trials we have open and available, you can do it just from your phone like you do everything else. And so you can pick by disease type, and you can get more and more specific if you know what you're looking for. But on the very front page of the app, it gives you a link to a phone number so that you can connect directly to someone in our office, or via email, so that we can really start to make that connection and put you in touch with the right people to figure out the next steps. Next slide. When we think about lots of different places for our patient advocates to get involved and to really partner with us, a big reason why we don't, let me go back. One thing that we would love to have from our patient advocates is help in designing better strategies for outreach and education in the community. It is very different to sit in my chair in the room versus in the individual patient's chair. And so what me or my team might think is a smart way to go about and do patient education may be totally wrong. And so having that kind of input from Pivot and our other advocacy groups is really essential to making sure that the right messages are getting across. We also, like Dan was talking about, and Kristi, when we're talking about ideas, it's really helpful to have patient voices coming in earlier in the process. And then also, as we think about research priorities and what's important to all of you, it could just help us identify where we should be putting our emphasis as well. Next slide. I think that's it. Great. Thank you.
Thank you so much, Dr. Lynn. Letty, do we have questions in the chat?
No questions.
Okie dokie. Then I will introduce Dr. Jennifer Klemp. And if some of you have been sitting for a while, I suggest you kind of shake things out a little bit, because we want to keep your attention till the end. I realize this is kind of rapid fire at you. So yeah, just kind of shake it out a little bit, wake yourself up. And Dr. Klemp, I will pass on to you. Are you ready?
Yes, thank you. And good to see all of you. And thank you for the opportunity to share a little bit about the Cancer Prevention and Control Program. Next slide. So this program has the mission, which I've put here, to reduce the burden of cancer and cancer disparities across the cancer control continuum. And so I wanted you guys to see, and just to kind of have a reference point, when we use that term cancer control continuum, it really means from primary, or people who don't have cancer, all the way through diagnosis, into treatment, and into survivorship. So our program deals with population-based studies of average risk people. It deals with individuals who are at high risk, and then it also deals with the survivor population. So if you click one more time on the slide, there you go. So as you just heard from Dan and Dr. and Tara Lynn, you'll see that right in the middle with diagnosis and treatment, those are sort of the two buckets that we don't sort of hyper focus on, but yet we still would be collaborative, because we do focus on survivorship on some of those quality of life issues that you heard, such as symptom management. Maybe it's something about patient-provider communication, genetics, you name it. We kind of cover the gamut, but we focus on sort of the beginning and the end. And as you can see, there's a lot of cross-cutting issues. And a lot of those have to deal with disparities, what's important across our catchment area, which Dr. Chen will talk with you about in the next presentation, and really focusing on things that we can do, targeting identification, getting people screened, how do we educate, and hopefully how do we mitigate or reduce that risk? We have 3 leaders. Dr. Ellerbeck is a preventive medicine physician, and he has a lot of experience in what we call implementation science, or how do you deliver something at the clinic and really make those workflows and the process better. He also has a lot of health services or larger scale research in rural health in kind of a bigger footprint, and a lot of work in cancer screening. Myself, I have some expertise in genetics and survivorship, as well as the use of technology and focus on quality of life. And then Dr. Nolan is a behavioral specialist who has a wealth of expertise in tobacco control and tobacco interventions, health disparities, and really engaging in community research. So it's very important for us to have kind of these complementary sort of skills so that we can hopefully sort of help guide some of the priorities and opportunities for the members of the program. And right now we currently have 49 associate and full members, but the cool part about the 49 members is they come from 23 different departments and 6 schools across the University of Kansas proper. Most of our members are from the KU Medical Center campus, but we also have members from both Children's Mercy and from the Lawrence campus. And I think the other really important thing that we're very proud of is over 70% of our members have national peer-reviewed funding. That could be from the National Cancer Institute, the Department of Defense. And so that's a really high mark. And it's great because we want our researchers to have those opportunities. And hopefully the program helps keep track of what the goals are from the catchment area or our community and our region to what funding opportunities are out there. We fund pilot research. And one of the other, I think, really strengths of our program is we have a lot of training grants. And those training grants allow us to have graduate students and train the next generation. So we often talk about that, but it's also very important to sort of walk the walk when you're talking about how do we empower and build the next generation of clinical researchers. Next slide. So that cancer control continuum is the thread that I want you to think about. And I think that's a really nice anchor as you guys get engaged more in a lot of these different levels and types of research. So the 3 types of research that we really focus on is population level, right? So this is where we really want to develop tests and innovative strategies that can modify behaviors before someone becomes at higher risk, or stop a behavior that's linked to cancer, like tobacco control, obesity, physical activity, immunizations, and cancer screening. So these are all very important. And as you all know, there's a variety of targets. There's probably, and Jill would be better at answering this question. There's probably, 12 different cancers that have a direct tie to obesity, and we have ties obviously to tobacco. So we want to kind of nip that in the bud. And there's a lot of our research focused on tobacco control. Another piece, when we get into high risk prevention, we want to work with our basic scientists, like what Dr. Dixon talked about in cancer biology. And we want to identify specific pathways, or how can we disrupt the cancer or put on the brakes, as you heard earlier as well. And we have researchers that have mouse models and human models, and they'll do a similar intervention between the 2. We have a researcher that is one of the world experts on Barrett's esophagus, which is a precursor to a type of cancer that happens sort of in the throat. And he's developed imaging techniques that are the standard of care. We have breast cancer researchers who focus on tissue sampling that allows us to see what's going on in the breast and hopefully disrupt that before it turns into cancer. So we're really focused on identification of high-risk populations through risk factors and genetics, and hopefully disrupting those pathways and those linkages to cancer. And then survivorship is a big, kind of umbrella, because survivorship by definition is from the time of diagnosis through the lifespan. So that means we have a lot of opportunity, not only once again in behavioral, nutritional, and clinical supportive interventions, but also how do we mitigate symptoms that a patient may develop during treatment, and how do we mitigate symptoms that they may develop 5 years, 10 years, 20 years down the road, like cardiac risk or bone loss, or the loss of fertility. And I think that some of our researchers who fall in this bucket, some of whom are on this meeting today, myself, Jill, Dr. Chen, we have this great constellation of folks who really prioritize that diagnosis through the lifespan and are trying to make the quality and quantity of life better for our patients and their caregivers with a diagnosis of cancer. So that's a really important piece as we continue to grow. And we have some growth in palliative care, as you also heard, with some funding that has recently come through, as well as some interventions that we're developing to try to better assess patients earlier so we can intervene and hopefully, once again, improve their quality and quantity of life. And then the last slide I wanted to kind of just quickly review with you is how do we overlap with Pivot and our community engagement and those priorities. So the first thing we do is we really want to focus on those top cancers in our catchment area, which are lung, GI cancers like colon cancer, breast cancer, and prostate cancer. We have our key factors, which we've talked about in regards to risk factors that we want to target, as well as health disparities, underrepresented populations where we're not doing the greatest job screening and getting them in for early detection and intervention. So a couple examples where we work with Pivot happen to be some of the projects that I work on, but I absolutely love working with Pivot. And I think, I hope I've established as an early adopter of the rapid reactor and other tools that are available. But one project in particular, Project BRA, I work with a medical oncologist named Lauren Nye. And what we've done is developed a educational and didactic program that works with primary care and other sort of boots on the ground providers across not just the Kansas City area, but across the state. And in doing so, we're teaching them how to do basic risk assessment right at the bedside and identify those patients at higher risk who may need genetic testing. Well, when we were developing some of the tools for Project BRA, including a genetic education video, not only did the rapid reactor with Pivot help us identify what context we should present the information, what should the individuals, and in our case, we used a high-end animation. What should that look like? What should be the speed? What type of roles should different people play? So we got so much amazing feedback, not only on the intervention itself, but also on the educational tools. And those are invaluable. So not only do we want to include individuals from Pivot early and to be able to understand the type of research and what our goal is, but then to hear from the Pivot members about how that really would work sort of from their vantage point, and how do we kind of merge the two so that our goals really are aligned. So I think the important part is that we do prioritize these partnerships. We absolutely depend on our members from Pivot, and we look forward to continuing those relationships as we build out some of these current and future research projects. And I look forward to getting to know you guys better. Hopefully we can see one another in person. And I want to thank you again for your time.
Thank you, Dr. Klemp, especially for showing an actual example where patients made a difference. Letty, are there questions in the chat before we go to our last speaker and then open up for questions?
No questions there.
I do have a
It looks
question
like there's
for
one
Dr.
question.
Klemp. Yeah.
Oh. Yes.
Thank you for your very wonderful presentation. And I do have a question about prevention and screening. For the breast cancer, the type I have, seems that two type of genome shows that statistically it's kind of ambiguous. And I never know, I'm the first one, and my family, we don't have any cases for breast cancer. So for me, I said, oh, should I do this? And should I do more? Tell my sister brothers to be careful. I don't know if I should stir the water. So
Right.
what's your advice for me?
So my advice is basically, without taking too much time, and we can always talk offline, is that I feel like cancer prevention aligns very nicely with cardiovascular prevention and diabetes prevention, right? We want to stay active. We want to keep a healthy weight. We want to avoid toxins like smoking and excessive sort of toxins to ourselves. So those messages, I think we should be sharing with every patient and everyone, even within our families. But then the next step is really doing a little deeper dive on the risk assessment. We also know that the Western lifestyle in and of itself is toxic and is a risk factor for cancer. So the perfect storm of cancer is that sometimes we don't have an answer, sometimes we don't have a genetic mutation we can test for, but all of us are at risk for cancer. So it's very important to share that message, to get screened early, and to really, even if we don't come from a high-risk family, we all have risk. And so the importance of outreach and screening are really vital. And that's where our Pivot members, in particular, some of you who I've worked with, including multiple people on this call, and if we don't get the village behind, these initiatives, it's really hard to achieve those goals.
Thank you, Dr. Klemp. Okay, and we will have discussion in a bit. We are going to wrap up with our final speaker, Dr. Ron Chen. Are you ready?
Yeah, thanks, Jill.
Okay, yeah.
Thanks, everybody. So I want to share a little bit about community outreach and engagement, which is a dedicated program at the University of Kansas Cancer Center. I'll share about what we do. Next slide, please. So the overall goal, I think, of the Cancer Center is to reduce cancer burden in the area that the Cancer Center serves. And specifically for the community outreach and engagement program that I help lead, there are several things that we do. One is the COE program. We help monitor the cancer rates in our area. And when we monitor cancer rates, we look for opportunities that we can then focus the Cancer Center efforts, whether it's in research efforts or outreach efforts. So we look for particular cancers that we can make an impact. I'll show you examples of exactly how we do this. We also, in community outreach engagement, we collaborate with communities in all of the efforts that we do. So we look for input. We also look for collaboration to help do as much good as we can related to reducing cancer burden. And also what we do in community outreach engagement is that we provide cancer screening and community education throughout our area. Next slide. So people may or may not know this, but the University of Kansas Cancer Center actually serves the entire state of Kansas, and also we serve 18 counties in Missouri. And part of the reason for this is because we're the only state cancer center, university-based cancer center in the state of Kansas, and therefore we feel that we need to serve the entire state of Kansas, even though we're located at the eastern boundary of the state. And because of our location, we also serve many patients in western Missouri. So when we talk about catchment area, or the area that we serve with the KU Cancer Center, we're serving this entire region. So we look for opportunities to collaborate with all the communities in this area, and we look for opportunity to serve all the cancer population in this entire area. Next slide. So, and this map shows the dots of where we actually have community collaborations and engagement. So we have multiple hospital center, cancer centers that are throughout the state of Kansas, also in western Missouri, that we formally partner with. And also the other thing that I think is really important as we form partnerships and collaborations is Pivot. We have over 100 Pivot members actively working with us, and Pivot members also are many dots on this map. So we really try to engage with communities and cancer members throughout the entire region so we can help get your feedback and also deliver intervention, education intervention to serve the community. Next slide. So one of the things that we do, as I said before, is we monitor the cancer rates in our catchment area. And of course, our catchment area includes both Kansas and Western Missouri. And some of the data that we monitor on a routine basis is we monitor the top cancers based on incidence, which is how many people develop or are diagnosed with the cancer. And we also monitor mortality rates, which is how many people die from these cancers. And so we monitor this for men, we monitor this for women. And so we look at these rates on an annual basis. The numbers that are highlighted in red are some of the cancers where the rates in our region are higher than the US overall. And so some of these red numbers where we have a higher incidence or mortality than the US overall may look like opportunities for us to make an intervention. Next slide. We also, as part of our data monitoring, we look at disparities. So we know that there are certain patient populations, whether it's by race or ethnicity or where people live, rural patients, for example, where there may be higher mortality because of cancer. So we look at disparities as well as another way for us to look for opportunities to make an impact. Next slide. And we also look at behaviors. There are certain behaviors that can potentially cause cancer, and there are certain behaviors where if we can intervene, we can reduce cancer. So we actually have data that we can monitor looking at smoking rates in our region, obesity, which is related to several cancers, and screening rates for colon cancer, screening rates for breast cancer, for example. And looking at these rates, we can also, if there's opportunities, for us to intervene to do education and increase screening so we can also reduce cancer incidence in certain areas. Next slide. So those are some of the things that we do in terms of monitoring data and look for opportunities. And we work with our community, we work with our community advisors on an annual basis to look for top priorities for the Cancer Center. And currently our top priority based on some of these data and feedback that we get from the community, we have a priority to address cancers related to tobacco. We have priority related to prostate cancer, breast cancer, GI cancer, and hematologic cancers, which are the blood cancers, leukemia, lymphoma. And also we have a new priority looking at obesity. These are some of the things where our community has felt that these are the important areas for us to focus our research and outreach efforts on. And we also have a priority related to impacting cancer care in rural areas where there may be underserved patients in those areas. Next slide. We do a lot of community outreach through the Masonic Cancer Alliance. We provide cancer screenings throughout our catchment area, and we've provided thousands of screenings throughout the catchment area. So that's something that we actively do to help make an impact in the communities. We provide a lot of community education. There is actually a Betch the Bedside, which is a Facebook educational series that's free for anyone to tap into, where we provide the latest advances and updates on research and other cancer care. And so we've impacted thousands and hundreds of thousands of individuals through this educational effort. We go out to swimming pools throughout the region to talk about the importance of sunscreens. We also provide a lot of education in community cancer centers to the providers and to the nurses so we can help educate that way as well. Next slide. And this is my last slide. Pivot is a really critical part of the community outreach and engagement program. And there are many ways how Pivot members can help us in this effort. I think primarily Pivot members help collaborate with researchers to drive research that will impact cancer in our catchment area. But also I think as Pivot members are living throughout Kansas, living throughout Missouri, you can also help us increase awareness locally in terms of cancer to promote screening events that we host. And you can provide feedback on what our priorities should be that may impact your local region. Thank you.
Thank you so much. We have now made it through all of the speakers.
Can I make a comment? Thank you very much. I think it's because of COVID, and nowadays we talk across nation, across the bound, to Taiwan and all that. And so your research here is not only impact local 2 states, it impact worldwide, in my view. Thank you so much.
Yeah, we hope to have an international impact as well. Thank you. That's great.
Thank you.
That's great. Well, I'm going to hand it over to Peggy Johnson. Peggy, you get to moderate the questions, so I'm going to go ahead and mute myself. Thank you.
Okay, thanks. I don't know about everyone else, but today has been great. short little snippets that just make us more interested in what's going on and we want to know more. And so I've got a whole bunch of questions here, but I'm going to look. I want to tell you one thing. Donnell has already downloaded the KU Cancer Center Clinical Trial Finder. Yay for you, Donnell. We've had lots of good information today. I'm going to start out with just asking one question, and then the rest of you, if you want to chime in or put it in the chat, it'd be great. But Dan, I'm going to ask you, just because drug development is something I'm interested in, and I'm especially interested in your team. Your chart showed drugs developed at KU and then drugs that are being repurposed. I actually know that I took a drug that was actually repurposed, which apparently worked. Can you tell me, the development of drugs takes so long and it's so expensive. When we repurpose a drug, is it a shorter discovery period and is it less expensive?
You're right on both of those takes, and I think Tara Lynn can attest to this also in when you actually get to the point of clinical trials with those drugs. So a lot of the testing in humans, the early testing, phase 1 and phase 2 testing, may have been done for a different type of disease, or just seeing how well that a person responds to that particular drug. And these would be done in normal people. And so that type of work really does not have to be justified. So we already know that information. So in a way you can skip that step with regard to these. If we know that a drug that's used for arthritis is safe in people, and now we want to test it in cancer patients, well, then we can take that safety question out and move right into cancer patients. And I'm simplifying this a lot. And Tara, if you want to add anything on to that.
I think you're spot on. So when we think about brand new drugs, they have to go through a phase of safety development where when they're first in humans, so the first time that we're testing them in humans, and that could take a couple of years to do. And so if we can use a drug where we already have some of that safety data, like Dan said, we can just leapfrog up further. And when we think about all the new things that we understand about cancer biology and just, medicines and everything in general, there are probably plenty of older drugs that we didn't know how to appropriately use at the time. Maybe we didn't test it in the right subtypes of this cancer or that cancer. So there's really a huge benefit if we can repurpose drugs that are already there and find that they can be effective as well.
That's great, thanks. I see that Donnell has put a question in it, and it says, what is the rate of skin cancers found in African Americans in our catchment area? What measures are we using to reach out to the community? So Dr. Chen, I'm going to send that one to you.
Dr. Chen let us know he did get a page from a patient. So Hope,
okay.
I don't know if you have that data, if you want to speak to that. If not, Danielle will definitely get that information from Dr. Chen and get it sent to everybody.
So the specific question related to the African American in our catchment area and skin cancer.
Yeah.
I think that we can get that. I don't believe that I have it off the top of my head. What we do is we pull information from the cancer registries every year and look at the skin cancer, at all the data by race, ethnicity. I might even be able to tell you it by the end of this call. Like I can go look it up right now and pull it from a database and give you that information. And as far as the measures for reaching out to the community, there's a variety of things that we're doing to look at that. And that's part of what we're actually asking our catchment committee. So we have an external group and an internal group called the catchment committee. It's made of, I think, 30 or 40 people. We look at lots of things. Some of them are just counting how many times, how many community members and what location are the community members from that we've actually touched, how many different community organizations are we interacting with, and who do they interact with as well. We look at how many projects we're doing that are specific to our like breast cancer or prostate cancer, et cetera. So we really look at by cancer site as well, and what are the outcomes of that. We report those in multiple ways too. So we are in this group often thinks about our NCI designation. And so we bring that information to our community advisory boards, our catchment committee, our leadership. We also report it to our funders, because there's funders that ask for this information. And then finally, we also have what's called a cancer committee for the University of Kansas Cancer Center. And that cancer committee is a clinical committee really focused on providing comprehensive cancer care. And so quarterly, we report to that cancer committee on our screening and prevention activities. And they also provide guidance on what we're doing and who we're engaging and how we're engaging. In addition to our outreach that are more individual focused, we also look at how are we interacting with healthcare professionals across the state, because we do feel like that 80% of people with cancer do not go to an NCI designated cancer center. So it's really key that we are looking at how are we also assuring that everyone has the same level of information and the same access to care. And then for clinical trials, Dr. Lynn and others are looking at, and in fact, we have a cancer care health delivery and equity committee that Dr. Chen and Jen Klemp and others are on, that are looking at race and ethnicity for geography and cancer sites, really looking at all those items for clinical trials accrual. So there's multiple measures. I'm happy to share a logic model if you all would like to see it at some time. And I think, and would also be really happy to share where we're doing well and where we're doing poorly, because we need you guys to know both the good and the bad so that you can help us be better. So that's a very quick answer of a pretty complicated question. But I appreciate the question, and I'll look up the skin cancer in Black populations, Dee, right now.
Thank you, Hope. I'm going to ask Danny a question, and there's a couple in the chat that I'll get to. But Danny, I know that immunotherapy wasn't actually tried in all cancers. So I know that it was more successful in some cancers than others. Could you just give us a brief explanation of how those cancers were selected? And I know that it's expanding all the time.
Well, so thanks, Peggy, for the question. And I'll just say I'm going to be sort of pulling some of this out of thin air as opposed to being directly involved. But a lot of the earliest cancers that were tried were types of cancer for which there have been more spontaneous regressions, where the patient's normal immune system actually attacked a tumor and they regressed. And those tended to occur most frequently in, say, renal cancers of the kidney or melanoma and the like. And so some of the earliest trials on the immune therapies were targeting some of those types of tumors because it's figuring tweaking the immune system is going to bump it up just a little bit. That's a bit oversimplified, but that's sort of there. In the last, say, 5 or 6 years, there's been an appreciation that instability, the genetic instability I mentioned pretty quickly in my What is Cancer, is a key determinant of whether some of these immune therapies are working. So when cancer cells are genetically unstable, they're generating all sorts of things that the immune system can attack. And the immune therapies, by and large, have been most effective against the tumors that are most genetically unstable. So there have been trials to identify which patients have the most unstable tumors, and they've been and using this the subset of patients that have that kind of observation. I know Tara and Ron and others probably can correct the details of that, but I think it's generally pretty close. I'm seeing Tara's head nod up and down, so I feel like I'm not too far off. I'd like to quickly just point something out for Danelle. One of the cancers that I study is melanoma, and one of the myths is that Black people or people of color don't get melanoma, and that's not the case. The real key is that where the cancers in the skin arise. So for the, and again, broad brush, because every cancer is different. They tend to arise between the toes or other places that are not highly sun exposed, because those are lighter in color. So the melanin is protective, and those kind of data do exist, but they're not in sort of the bigger database, but there are people on our campuses studying it, so.
That was why I asked too, because I know that there's a myth out there. And so if our African-American community can be touched by other African-Americans who have experienced skin cancers, it will help with the screening and prevention.
Sure.
Yeah, and Danelle, I'd say it's not, the myth also needs to be addressed among healthcare professionals too. And there's been a lot of research recently, or a lot of information lately about just medical care in general, and really understanding how different diseases present with different skin tones. And so I think that medical schools are now trying to do a better job of that in general. Just for everyone to know, for skin cancer, when it's for the cancer registries, they only, and I should have said this at the beginning, they only look at melanoma. So it isn't going to have any of the other skin cancers. And the reason they look at melanoma is the one that's going to most likely become metastatic of the skin cancers.
Yeah.
So there's another question in the chat, and I honestly don't know who to put this to. So any of you chime in. Has there been any research on research and alcohol and cancer? And I know, we don't, in breast cancer, we don't say causes cancer, but we say that it is a lifestyle that could affect your breast cancer, and alcohol is one of them. So I'm at a loss of who to ask.
I would throw it to Kristi.
Yeah.
Neufeld.
Yeah, you can throw it to me. We do have a big group of researchers studying liver and liver biology, and I know that some of them are studying the linkage between alcohol consumption and sort of precursor stages to cancer, and how that might evolve into cancer. So there are people, researchers on in the Cancer Center working on that connection. I think in general, if I'm remembering from, I teach a cancer biology class, and I think I had this on a slide, alcohol is associated with maybe 6%, or thought to be estimated to be associated as a risk factor for about 6% of all cancers are attributed to alcohol consumption. So there's very likely a connection, and yeah, some of our research are studying that connection.
And Kristi, that's scary, because if you think about the pandemic where more people drank more and screened less,
yeah,
that's a scary idea.
I think
and there
the
are
other
some
thing,
people who are studying it every Friday night, but from a different perspective. There are a number of studies, I think, collaboratively. Just this is off target a little bit, but I'm collaborating with someone at my former institution, UAB. And there are differences in races and how alcohol is processed. And that does tend to seem to correlate with changes in not only liver cancer, but some of the head and neck tumors that are highly alcohol associated. So more details at 11, but those are very early.
Dan?
Yeah, I was just going to say the research that's been done, especially in like breast cancer, like you alluded to, Peggy, they look at the number of ounces per day, but the challenge is that's not how people intake alcohol. So you may have like Friday night, a couple glasses of wine, but it looks at, 6 ounces per day. And so there's a limit per day. So the research and how people actually utilize the behavior aren't really consistent. But there is a link to increasing cancer, like Danny said, with liver and head and neck, breast, a variety of cancers.
Yeah.
Okay.
Great. So actually, sorry, Jen, or Peggy, that what Jen was just saying about alcohol consumption and how it's consumed. I'm going to come back to Dee's since you've renamed yourself. Question about the skin cancer. It's very interesting. If you, one of the questions I often pitch to grad students on the first day is, who do you think job-wise is most susceptible to getting skin cancer? And it's dentists. And it's because dentists work throughout the week, although they seem to be working less and less days a week based upon my ability to get an appointment. It tends to be the people who go out and binge in the sun, as opposed to farmers and things like that are out in it all the time. So the sophistication by which we look at all these causes has got a lot of layers, and we need to take that into account.
Thank you, Danny. So it's 1:30. You all have been great. Actually, I think this has probably whet your appetite for more information on all these things. The thing I want to remind you is that we got a PDF of all the slides today in our Zoom connection. So I encourage you to download those. I know it's been recorded. I'm going to go back and listen again. I was trying to take notes, and so I really need to go back and just listen for pure informational. And I really want to plug Bench to Bedside. If you have not done or been able to sign in and do a Bench to Bedside, I know Tracy's doing two, and then in the future, I did one a year ago. I went back and looked at it about 2 weeks ago just to check my hair, see what it looked like. But it was a great session. They are all available on YouTube. So if there's a specific topic that you would like to go back and revisit, they're out there. So please go back, use Bench to Bedside. I can't see them on Wednesday. I have a 2-year-old that just doesn't like me to look at the computer, but it's a grandson.
Can I jump in really quick? As she said, I am doing 2 Bench to Bedsides, one November 3rd about pelvic floor therapy. It's what happens sometimes to men and women after chemotherapy. And then December 3rd, it'll be me and my researcher talking, the one that we are working together with on a trial.
Fabulous.
Sorry, Peggy.
No, it's all right. It's all right. I'm looking to see, it's great. I want to thank the people on the Education Task Force. I think we're all on here today. They did a lot of work putting these together, but it turned out well. Please do us tomorrow. We got one more to do tomorrow, and it's at noon. And so I want to especially thank our speakers today. So Dr. Chen, Dr. Dixon, I'm looking to see everybody. Tracy did one. I'm going back and look at my notes. You know who you are. We appreciate it. Danny, you did a great job. Kristi, a great job. Please reach out to these people if you have more questions, and we appreciate everything that you did today. I'm seeing lots of hand claps, so very good. Very good, yes. Jill, I'm going to turn it back to you.
Okay, well, I won't take much time here. I look forward to seeing all of you on Friday. And thanks for coming back today. And thanks for helping us stay on time. Thanks to the speakers. Take care. Have a good day.
See you tomorrow.
Bye-bye.